In 2017, inside a sleep lab at the University of Wisconsin, a research team sliced open a mouse's brain in the middle of the night and found that nearly 1 in 5 of its synaptic connections had vanished. Not damaged, not dormant, gone. And neuroscientists still cannot explain how the brain decides exactly which connections to erase. Picture the lab at 3 a.m. The mouse is asleep, tucked under soft red light so the researchers can watch without waking it. Electrodes trace its brain waves. Somewhere behind the University of Wisconsin's Department of Psychiatry, Giulio Tononi and Chiara Cirelli had spent over a decade chasing a single question: why do we sleep at all? Their answer would come from cutting into thousands of synapses, one microscopic connection at a time, and counting. The first anomaly showed up under an electron microscope. Synapses photographed after a full day of waking activity were dense, swollen, packed with receptors. Synapses photographed after a night of deep sleep had shrunk, on average 18% smaller. Some had disappeared entirely. This wasn't isolated to one brain region. It happened across the cortex, night after night, animal after animal. Your brain, it turned out, spends its waking hours accumulating connections and spends its sleeping hours deleting them. Billions of them, every single night. The obvious theory arrived fast, and it sounded almost comforting. Maintenance. Cleanup. A biological reset button, clearing out clutter so the brain doesn't overload on structural growth day after day. Tononi called it the synaptic homeostasis hypothesis, sleep as a nightly bill collector, downsizing the whole system back to baseline. Simple, mechanical, the kind of tidy answer that lets you stop asking questions. Except the shrinkage wasn't uniform, and that's what breaks the theory. If this were random downsizing, pure maintenance, no intelligence behind it, every synapse should shrink by roughly the same amount. They didn't. Some synapses, the ones tied to a memory the animal had just formed that day, were left almost untouched. A few were even strengthened overnight. Meanwhile, synapses carrying old, unused information were stripped down aggressively, sometimes eliminated outright. Something inside the sleeping brain was making a selection, reading which connections mattered and which didn't without the animal being conscious, without any input from the very memory being judged. Here is the detail that still keeps researchers up at night, no pun intended. In 2020, a follow-up study out of the same lab used a genetically engineered marker to watch astrocytes, support cells once thought to be neural janitors. Nothing more physically engulfed specific synapses during slow-wave sleep. Not synapses at random, specific ones. The astrocytes appeared to be executing a targeted removal, like a demolition crew working from blueprints nobody had drawn. And remember that 18% figure from the opening scan. Researchers still cannot identify the signal that tells an astrocyte which synapse is expendable and which one is a memory in progress. There is no known molecular tag, no confirmed, marked for deletion flag, that fully accounts for the selectivity. The brain is editing itself with a precision nobody can locate the source of. Competing explanations have piled up, and each one collapses somewhere. Some researchers point to local protein synthesis, the idea that recently active synapses get chemically protected by proteins built on the spot. It explains some preservation, but not the aggressive, selective pruning of the unused connections. Others blame the glymphatic system, the brain's overnight waste-clearing plumbing, discovered in 2012. But that system flushes metabolic waste, not synaptic architecture. It doesn't explain why specific memories survive untouched while others get erased outright. Hormonal signaling has been proposed too, tied to slow-wave sleep's growth hormone pulses, but the timing doesn't line up cleanly with which synapses disappear. Every theory accounts for part of the picture. None of them account for the selection itself, the mechanism deciding what you get to keep. And there's a darker implication sitting underneath all of it. If your brain is making this selection every night, on a schedule you have no conscious access to, then somewhere in your skull, something is quietly deciding which version of your past you get to wake up with tomorrow. People with fragmented slow-wave sleep, shift workers, insomniacs, Some dementia patients show measurably worse memory retention, as if the deletion process itself starts malfunctioning, taking the wrong things. Nobody can say why. Nobody can say what happens when the wrong synapse gets marked. So tonight, while you sleep, that same unexplained process starts again. And nobody, not Tononi's lab, not anyone since, can tell you exactly what it's about to take from you.